Good afternoon, everyone. I'm final year PhD student in the medicinal chemistry lab in the School of Chemistry and Molecular Bioscience at the University of Queensland. And today I'm going to present on my PhD project And this project is supervised by Professor Toth and Dr. Marias. Well, gram-positive bacteria, group A streptococcus is a gram-positive bacteria responsible to cause several invasive and non-invasive complications. The most common complication of group A streptococcus is rheumatic heart disease. And in 2010, it was estimated that 70 million people around the world has been suffering from this disease. And this disease potentially creates 1.4 million deaths a year. And usually, group A streptococcus disease infection spreads by direct contact of bacteria with mucus or secretion from an infected person. Although this organism is fairly susceptible to antibiotics, currently available methods of treatments are often inadequate, particularly in the developing country settings where this disease is continuously re-emerging even after the cessation of therapy. Therefore, early prevention against this group A streptococcus is required. And unfortunately, there is no gas vaccines available in the market. So if we see the current overall status of current available vaccines, we see most of the vaccines are delivered by parenteral mode of administration. And there are very few vaccines that are administered by oral route. And most of the vaccines that are prepared for oral routes are prepared from inactivated or live attenuated organisms. And as we can see, there are no peptide-based vaccines in the market. Although oral vaccines have several potential advantages, particularly due to their simplicity in administration and self-delivery possibility of self-delivery, and they have a advantage of reducing the risk associated with the needle-based injury, and they also have a potential for the mass immunization. The most important part of any mucosal vaccines is that they have a capacity to induce mucosal response as well as systemic immune response. However, oral vaccines development has been hampered by several gastrointestinal barriers, particularly barrier in the gastrointestinal tract. As we know that protein or peptide-based vaccines, they need to expose to the low acidic pH in the stomach, and it needs to survive the presence of proteolytic enzymes. So even though they manage to survive this environment, when they reach to the intestine, they may not be sufficiently absorbed due to the high molecular weight or too hydrophilicity of the antigen. Since our lab has, since a few years, our lab is extensively working with the lipid core peptide-based vaccines. These vaccines usually consist of lipid core peptides, which is analogous to non-microbial lipopeptide adjuvants. These are conjugated to multiple peptide epitopes with a lysine linker. And this whole system is amphiphilic with a hydrophobic and hydrophilic. And the most important feature of the lipid core peptide vaccine is they can be easily synthesized in the lab using basic chemistry. And they have almost more than 95% pure vaccine candidate can be synthesized. This is one of the most important vaccine candidate that has been synthesized in our lab, and it has shown promising against group A streptococcus when they are delivered by subcutaneous and intranasal route. Basically, it contains two C16 lipoamino acids in the core, and it is conjugated by the linker and attached to the multiple epitopes. One is CD40 helper epitope, which is P25. This is universal CD40 helper epitope, and the other is B cell epitope. And this is B cell epitope is derived from M protein, and this is the minimum peptide sequence that is derived from M protein. And M protein is the most virulent factor for group A streptococcus. Previous experience with the gas vaccine for the oral administration was not promising. As we can see, this J14 peptide, which is B cell epitope, when they were conjugated with a heteropolymer system and delivered orally, even with the co-delivering with cholera toxin and through feeding them with the alkalizer sodium bicarbonate, and despite of extensive immunization, they were unable to show for any specific response. And later, another peptide, which is also active against group A streptococcus called J8, which was conjugated to LCP, had shown a bit of promising result, but this was significantly lower than as compared to intranasal route. And they also used 7 oral boosts to get this response. So we assume that this could be due to the substantial degradation of purified epitopes from the gastrointestinal system. So that makes them, unstable in the GI tract, making the less antigen to be available for the uptaken by the antigen presenting cells. So we hypothesized that why not use a proper delivery system based on nanoparticle, so that we can modify and protect the entire structural integrity of the antigenic vaccine component and deliver through the gastrointestinal system using oral route. So we decided to design a new delivery system based on polyacrylate coating. So as I already told you that our vaccine candidate, also named as lipopeptides, it contains 2 hydrophilic and lipophilic structure. So using the self-assembly behavior of this and vaccine candidate and cationic phospholipids, we loaded lipopeptide into the liposomes. And these liposomes were nano size, particularly around 160 nanometer. And this LCP loaded, vaccine candidate loaded nanoliposomes were later coated with a different series of oppositely charged polymer. First, it was sodium alginate, and sodium alginate is enteric acid resistant polymer and has a good solubility at intestinal pH. When they reach the intestine, it has a good solubility. And trimethyl chitosan is a positively charged polymer, which is a modified form of chitosan, and it has a M cell internalizing property plus enhanced permeation capacity from the intestine. So we coated this with both the polymer using electrostatic interaction technique, and we prepared series of different layered liposomes. So the idea is when these multi-layered liposomes are administered, so when they reach to the stomach at low acidic pH, the outer core with alginate will protect, since this is enteric acid resistant polymer, and when they slowly reach to the small intestine, this outer layer erodes and exposes with the trimethyl chitosan, and since this is mucoadhesive, we expect them to be absorbed through from this intestinal membrane to be presented to antigen presenting cell. So we first started to optimize the formulation ingredients. We defined how much concentration of phospholipids are required to prepare safe, stable liposomes, and we found that DPPC is a phospholipid, cholesterol, and another phospholipid at a concentration of at a molar ratio of 2:1 to 0.01 produce the base liposome with 157 nanometers in size, and DDAB was particularly added to the liposomes to impart a positive charge. So it was important for us to optimize the exact concentration of polymer to coat to the liposomes, because we don't want excess polymer. We don't want them to generate extra immune response or unwanted response. So we precisely optimized them, and we found that for first coating, we found that this concentration will be enough, and second coating, this amount is enough. Similarly, this coating was enough. So we optimized all the required component and prepared cell-stable polyacrylate nanoliposomes, coated nanoliposomes. So to summarize, our vaccine candidate was highly aggregating with 171 nanometer size. So this blank liposome without LCP was 157. And so when it was incorporated into the nanoliposomes, it produced a bit higher particle size. So upon adding each layer, particle size slowly increased. As you can see, there was a 5 nanometer increment with the first layer, and there is 12 nanometer increment with the second layer coating, and almost the 30 nanometer increment upon the coating third layer. And all the developed formulations were monodispersed in site. And the evidence of coating can be shown by the change in zeta potential, that is surface charge of the in the particle. Here you can see positive and negative, and again positive and negative. And this size was later verified with the transmission electron microscope image. And as here we can also see they were still able to maintain the spherical morphology when they are even dried. So we decided to see whether these developed formulation systems had enough stability in the simulated gastrointestinal fluid under in vitro condition. So we prepared different simulated gastric fluid and simulated intestinal fluid and incubated them for 4 hours, 2 hours into the simulated gastrointestinal fluid and 4 hours in the intestinal fluid. And we measured the particle size and their dispersity, and we found that they were still able to, formulation when LCP was, only LCP were aggregating when it was in the pH, high intestinal pH. However, when loaded into the different formulation system, they were enough stable, particularly in pH 1.2 and however, at intestinal pH, that is around 6.8, we found that there was a slightly significant increment in particle size. But this was not surprising to us because alginate has inherent capacity of swelling when they are exposed to the intestinal system, intestinal pH. So after this, we wanted to see whether this developed formulation had a capacity to be taken up by antigen-presenting cell, and splenocytes were harvested, and our formulations were incubated into the splenocytes, and these were tagged. The dendritic cell and macrophage were tagged. We studied the capacity of these antigen-presenting cells to uptake our formulations, and we found that most of the positively charged liposomes, which has a trimethyl chitosan outside, had a significantly higher uptake as compared to the negative charge liposome. For alginate, which is covered with alginate, is negative charge. They had significantly higher uptake, and same trend was observed with the macrophages. And this could be because we believe that because negative charge on the particles was unfavored to be taken up by the negative surface charge of the cells. So as we know that antigen-presenting cells continuously take up foreign particles from the environment. So high uptake always doesn't guarantee that immune cascade will occur. So that our formulation system or our delivery system can provide extra danger signal for the immune cascade to occur, we decided to go with animal study. So animal study was conducted with the Swiss outbred mice, and which is 6 to 7 weeks old, and we orally gabas mouse with 100 microgram of LCV equivalent of LC vaccine, which were loaded into the formulation, and this was make up to 200 microliters. So we used 2 sets of positive control. One was with mucosal adjuvants cholera toxin, 10 microgram cholera toxin. The other one is with 60 microgram, and this was already proved and we already published that it was working well, so we use this as a positive control, these 2 as a positive control. And all these animals were administered gavas without anesthesia, even intranasal. So this is general timeline, and we did 3 boosting and collected sera, and we did, after 5 boosting, we collect the sample, and we wanted to see if they have a persistent long-term immunity, and up to we case them up to 6 more months from the starting, and we re-stimulate them and collected the sample. So after 3rd boosting, we found that LCP, which is our main vaccine candidate, they were not stable, and they were not producing any immune response apart from 1 mice. And when they were loaded into the liposomes, there was a slight increment in the immune response, in the systemic immune response, however, not significantly higher. And all these formulations which produce, with even this physical mixture, were able to produce a bit higher response as compared to the LCP alone. And if there is interesting, and upon coating with just 1 layer with the negative charge alginate, there was no much change as compared with the uncoated one. But with the coating with the trimethyl chitosan, it significantly improved. And improved, and this was one of the formulation that, and this was even comparable to our positive control, which was oligobas with CTB and when an LCP, which was administered intranasally. So it is really nice data to show that coating is really helping us to improve the immune response. And we decided to see whether they have IgA response or not. And as we can see here, again, we obtained a similar trend. And this formulation, which is a positively charged double layer liposomes, which has a trimethyl chitosan, and the outer core had a significant uptake as compared to PBS and compared to this LCP alone. And these two lead vaccines, this lead vaccine candidate was taken off, was orally immunized with 2 extra boosts, and we found they were almost similar and showing the similar trend, but there was no change in IgA level, but there was slight increment in the IgG upon additional 2 doses to this. So this result implies that additional 2 doses was not that actually required and was enough. 3 doses was enough when we delivered. And we decided to go with the long-term response, and we found that there was, before re-stimulating them with this same formulation, there was still an immune response, and upon re-stimulation, there was a slight increment, but not so much. And similar trend was obtained for IgA response with the salivary trend. So in conclusion, we developed a new delivery system for oral administration of peptide-based vaccine with excellent property. I forgot to say that we had a high loading efficiency of LCP into our formulation system with more than 80%. And all our formulations showed enhanced stability in this gastrointestinal fluid, and they were uptaken by antigen present in cell. And they were even thought 3 boosts were sufficient to enable efficient immunity, and was able to show long-lasting immunity. So in conclusion, our results suggest that layer-by-layer engineered nanoarchitecture formulation as a promising novel therapeutic strategy for peptide-based oral vaccination. So finally, I'd like to acknowledge all this person involved. And we did animal study in Griffith, and I'd like to acknowledge Professor Michael Good and Michael Baslov and my colleagues, and funding body for my PhD study and all project funding body. Thank you.
Okay, we have time for some questions. So in terms of bile salts, how stable are these? Did your stimulated gastric secretions contain high levels of bile salts?
Yeah, I showed you one data with the in vitro stability. We simulated with the bile salt as well. So there is a recipe which I referred from the pharmacopoeia, and it incorporates bile salt together with all these pancreatic enzymes and everything.